Welcome to another episode from the Veterinary Medicine Digest. Today, we're scratching the surface of a topic that plagues countless pets and their families. The relentless, maddening sensation of itch. We're talking about pruritus, the medical term for a sensation that provokes the desire to scratch. For any pet owner who has watched their beloved dog or cat chew, lick, and scratch themselves raw, you know this is far more than a simple annoyance. Pruritus is a cardinal clinical sign of many skin disorders, and it is currently the single most common complaint in canine dermatology. The constant discomfort can profoundly impair the quality of life for our animal companions, and by extension, for us, their dedicated caregivers. In this episode, we're going to take a deep dive into the world of veterinary itch. We'll explore what it is and what causes it on a fundamental biological level. We'll peel back the curtain on how scientists are creating sophisticated models to study this complex sensation and what those models are teaching us. We'll also look at the cutting-edge techniques that are revealing the hidden changes within the immune systems of itchy pets. And finally, we will walk through the modern arsenal of treatments, from traditional standbys to revolutionary targeted therapies that are bringing relief to millions of suffering animals. So, settle in as we unravel the intricate science behind the scratch. To begin, let's define our terms. What exactly is itch? On the surface, the definition seems obvious. It's a feeling that makes you want to scratch. In its most basic form, this sensation is actually a protective reflex. When an insect, a chemical, or a poisonous plant comes into contact with the skin, the itch sensation serves as a first line of defense, prompting the animal to remove the irritant. This is what we would call acute itch, and it's a useful survival mechanism. But when itch becomes chronic, the story changes. The International Forum for the Study of Itch defines chronic itch as any pruritus lasting longer than 6 weeks. In our veterinary patients, this persistent, long-term itch is rarely a simple, one-off reaction. It's usually a sign of a deeper, ongoing inflammatory skin disease, most often rooted in hypersensitivities or allergies. The way our pets express this discomfort can vary dramatically. Some signs are obvious, like frantic scratching, rubbing against furniture, or incessant licking of their paws. But other signs can be much more subtle, such as the gradual development of hair loss from overgrooming, a condition known as self-induced alopecia, especially common in our feline friends. Behind this seemingly simple behavior of scratching lies an incredibly complex pathophysiology that we are still working to fully understand. The challenge is even greater in veterinary medicine because our patients can't tell us what they're feeling. We assume that what a dog or cat experiences as itch is similar to the human sensation, which is often a mix of not just itching, but also burning, prickling, or stinging feelings. This chronic discomfort can seriously impact an animal's quality of life, leading to anxiety, sleeplessness, and distress. So, what is happening under the skin to create this tormenting sensation? For a long time, scientists have known that the pathways for itch and pain in the nervous system are closely related. This explains a common phenomenon. Scratching, which creates a mild pain signal, can temporarily relieve the sensation of itch. However, research has revealed that there are also specific neuronal pathways dedicated solely to itch, distinct from those that transmit pain. There are 2 main neurophysiological pathways that can transmit an itch signal from the skin to the brain. The first is a histamine-dependent pathway. This is the classic itch pathway we associate with things like mosquito bites or hives. An insult to the skin causes specialized cells, like mast cells and keratinocytes, to release a flood of histamine. This histamine then binds to its receptors on specific, sensitive nerve fibers in the skin, sending a direct itch signal to the brain. The second pathway is independent of histamine and is much more relevant to the chronic itch we see in allergic pets. This pathway is activated by a whole host of other itch-inducing substances, or pruritogens. The itch signal starts when these substances stimulate specialized nerve endings in the skin called pruriceptors. From there, the message travels along unmyelinated nerve fibers to the spinal cord, then up a pathway called the spinothalamic tract to a region of the brain called the thalamus. Ultimately, the brain interprets this signal and generates a motor response, the physical act of scratching. In the last decade, one particular pruritogen has stolen the spotlight in both human and veterinary dermatology, a cytokine known as interleukin 31. Cytokines are small proteins that act as messengers between cells, and interleukin 31 has emerged as a master conductor of allergic itch. It belongs to the interleukin-6 family of cytokines and is mainly produced by a type of immune cell called a T helper 2 cell, which is heavily involved in allergic reactions. When interleukin 31 is released, it binds to a specific receptor complex found on the surface of sensory neurons in the skin. This binding event triggers a chain reaction inside the neuron, a signaling cascade known as the Janus kinase signal transducer and activator of transcription pathway, or JK-STAT pathway for short. The activation of this pathway is what ultimately generates the itch sensation. The evidence for interleukin 31's central role is overwhelming. Scientists have shown that injecting recombinant interleukin 31 into mice, dogs, primates, and even humans causes intense itching behaviors. Even more compelling is the success of modern therapies designed to block this pathway. Medications like locavetimab, which is a monoclonal antibody that specifically neutralizes canine interleukin 31, have been remarkably effective at reducing itch and inflammation in dogs with atopic dermatitis. This powerful evidence confirms that interleukin 31 is a major, and perhaps the most significant, pruritic player contributing to the misery of allergic itch in our canine companions. Understanding these molecules is one thing, but to develop and test new anti-itch medications, researchers need reliable and repeatable ways to study pruritus in a controlled setting. This brings us to the fascinating world of canine itch models. One of the most important tools in this field is the canine interleukin 31 induced pruritus model. This is where researchers can safely and temporarily induce itch in healthy dogs to evaluate how well new drugs work. A landmark study published in the journal Veterinary Dermatology sought to deeply characterize this model to make it more accurate and useful for future research. The study's primary goal was to get a detailed picture of the immediate and delayed itch responses after an interleukin 31 injection, and to see if the response was consistent when the injection was repeated. The researchers used 15 healthy, adult male laboratory beagles. This was a randomized, controlled crossover study, which is a very robust scientific design where each dog serves as its own control, minimizing the influence of individual variations in how they might react to the itch. Each dog received 4 intravenous interventions in a random order, 2 injections of recombinant canine interleukin 31 and 2 injections of a placebo vehicle, which is a simple phosphate-buffered saline solution. There is a 14-day washout period between each injection to allow the dog's system to return to normal. After each injection, the dogs were video recorded continuously for 270 minutes, or 4.5 hours. 2 trained investigators who were blinded so they didn't know which injection a dog had received painstakingly reviewed every second of these videos to score the dog's pruritic behaviors. These behaviors included everything from licking and chewing to scratching, head shaking, and scooting. One of the most crucial findings of this study revolved around how the itch was measured. Historically, many studies used a simple categorical scoring system. For each 1-minute interval, the observer would just mark yes or no for whether any itching occurred. The researchers in this study hypothesized that this method was not very precise. For example, a dog that scratches for just 5 seconds in a minute would get the exact same score as a dog that scratches for 50 seconds. This could hide the true severity of the itch and potentially bias the results of drug trials. So, in addition to the old method, they also measured the total duration of all pruritic behaviors in seconds. The results were striking. As expected, the interleukin 31 injections caused a significant increase in itching compared to the placebo, with some dogs showing up to a 28-fold increase in pruritic behavior. This powerfully reaffirmed that interleukin 31 is a key driver of itch in dogs. But here's where it got really interesting. The second interleukin 31 injection, given 14 days after the first, induced a significantly greater amount of itching than the first one did. This suggests that the first exposure to interleukin 31 might sensitize the dog system, perhaps by causing an upregulation of the interleukin 31 receptors on neurons, making them more responsive the next time. This is a critical finding for future research, as it suggests that a simple 2-week washout period might not be long enough between challenges. And here is the methodological bombshell. The more precise total seconds scoring method clearly detected this significant difference in itch intensity between the first and second injections. In contrast, the old yes/no per minute scoring system completely missed it. According to that method, there was no statistical difference between the 2 injections. This was a clear demonstration that the categorical scoring system lacks the sensitivity to accurately reveal the severity and duration of itch. The study's authors strongly concluded that all future evaluations of antipruritic drugs using this model should assess total itch behavior in seconds to avoid biased and inaccurate results. The study also shed light on the timing of the itch response. The pruritus wasn't immediate. In fact, there was no significant difference in itching between the interleukin 31 and placebo groups in the first 30 minutes after the injection. The real itch kicked in later, showing a delayed response that peaked between 120 and 210 minutes post-injection. This delayed onset is similar to what is seen in humans and rodents and suggests that interleukin 31 doesn't just work by directly zapping a nerve. Instead, it likely initiates a more complex cascade, possibly by stimulating skin cells like keratinocytes to release a secondary wave of other pruritogens that then amplify and sustain the itch signal. The study also documented that the most common behaviors induced by the interleukin 31 were scratching, head shaking, and biting or chewing. Furthermore, the itchy dogs slept significantly less, highlighting the restlessness and diminished quality of life that pruritus can cause. While this model gives us incredible insight into one specific itch pathway, what's happening in the bodies of real-world pets with chronic, naturally occurring allergies? To answer this, another group of researchers employed a powerful technology called hyperammeter immunophenotyping. This technique uses an instrument called a spectral flow cytometer to analyze individual cells in a blood sample, identifying and quantifying many different types of immune cells at once based on unique markers on their surface. The researchers in this study took it a step further by using sophisticated machine learning and unsupervised clustering algorithms to analyze the complex data. This objective, computer-driven approach can identify distinct cell populations and uncover subtle patterns that might be missed by a human analyst, all without preconceived notions or bias. They collected blood from a large group of client-owned and laboratory animals, including 55 dogs and 68 cats. These animals were categorized as either pruritic if they were showing active signs of itch, or non-pruritic if they were in for a routine checkup. The results revealed fascinating and distinct immune profiles in itchy pets, with some surprising differences between the two species. Let's start with the cats. When looking at a standard complete blood count, the pruritic cats showed a classic picture of allergic inflammation. Their total white blood cell count was significantly higher than in non-pruritic cats, driven by a significant increase in two specific cell types, eosinophils and neutrophils. Eosinophils, in particular, are strongly associated with allergic reactions, and this finding was confirmed by both the standard hematology analyzer and the advanced flow cytometry analysis. But the hyperammeter analysis uncovered much more subtle changes. Pruritic cats showed a significant shift in their T-cell populations, the master regulators of the immune response. Overall, they had fewer circulating CD4 positive T cells, a key type of helper T cell. More specifically, they showed a significant decrease in subsets of both CD4 positive and CD8 positive T cells that were CD62L high. CD62L is a surface molecule that acts like a homing beacon, keeping naive T cells circulating through lymph nodes. When T cells become activated, they often shed this molecule, allowing them to leave the bloodstream and travel to sites of inflammation, like the skin. So a decrease in circulating CD62L high T cells in pruritic cats suggests a systemic shift towards a more activated or effector T cell population that is migrating out of the blood and into the skin to perpetuate the allergic inflammation. This aligns perfectly with previous studies showing that cats with allergic dermatitis have an increased infiltration of T cells in their skin lesions. The story in dogs was quite different, highlighting that we can't simply extrapolate findings from one species to another. In stark contrast to cats, the pruritic dogs had significantly lower total white blood cell counts than the non-pruritic dogs, primarily due to a decrease in neutrophils. The changes in their T-cell populations were also the opposite of what was seen in cats. Pruritic dogs had a significant increase in the proportion of circulating CD62L, high CD8 positive T cells. This might indicate a clonal expansion of newly activated T cells that are still present in the circulation. The analysis also revealed unique subsets of another immune cell type, the monocyte. In dogs, the researchers identified 3 distinct monocyte populations, and they found that pruritic dogs had a significant increase in the proportion of a specific subset called CD4 positive monocytes. While we normally think of CD4 as a marker for helper T cells, it is also expressed on some myeloid cells in dogs, and an increase in this particular monocyte subset may be an indicator of a systemic inflammatory state. This study's powerful combination of spectral flow cytometry and artificial intelligence analysis demonstrates that there are profound, measurable differences in the circulating immune cells of pruritic animals, and these immune signatures differ between cats and dogs. This work opens up exciting new avenues for identifying disease biomarkers and developing more targeted therapies. With this deeper understanding of the mechanisms and cellular players involved in itch, we can now turn to the practical question: how do we treat it? The modern approach to managing pruritus is a multifaceted one, involving treating the underlying cause whenever possible, along with topical and systemic symptomatic therapies. A comprehensive review published in Veterinary Sciences provides an excellent framework for choosing the right antipruritic drug for the right situation. Let's start with the old guard: glucocorticoids or steroids, such as prednisone and prednisolone. For decades, these have been the go-to drugs for controlling inflammation and pruritus. They are incredibly effective for managing acute flares of atopic dermatitis, and they work fast, providing relief within hours to a few days. However, their powerful effects come with a significant list of potential side effects, especially with long-term use. The most common short-term side effects are what veterinarians call the big 3: polyuria, increased urination; polydipsia, increased thirst; and polyphagia, increased appetite. While manageable, long-term use is generally not recommended because it carries more serious risks, including suppression of the adrenal glands, an increased susceptibility to infections, and the potential to induce diabetes. Therefore, systemic steroids are best thought of as a fire engine, excellent for putting out the fire of an acute flare-up, but not for long-term fire prevention. Topical steroids like hydrocortisone acetonate spray are a safer option for localized lesions. They can be very effective and have far fewer systemic side effects, although prolonged use can still lead to localized skin thinning. Proactive use of a topical steroid spray on previously inflamed areas a couple of times a week can even help prevent relapses. Another class of older drugs is the antihistamines. These medications work by blocking histamine receptors. Unfortunately, as we discussed earlier, the chronic itch of atopic dermatitis is largely driven by non-histaminergic pathways. As a result, the clinical efficacy of antihistamines in dogs is modest at best and often unpredictable. They might be worth a try for very mild cases or as part of a multimodal approach to see if they can help reduce the required dose of more potent medications, but they are not recommended as a sole therapy for significant allergic flares. Their most common side effect is sedation. A more powerful tool for long-term management is cyclosporin. This drug is an immunomodulator that works by inhibiting T cells from producing inflammatory cytokines. It is a potent and effective antipruritic medication, but its major drawback is its slow onset of action. It can take 4 to 6 weeks of daily administration to see the full beneficial effect. This makes it unsuitable for controlling acute flares, but an excellent choice for the long-term, proactive management of chronic atopic dermatitis. Once the disease is under control, the dosing frequency can often be tapered down to every other day or even twice a week. The most common side effects are gastrointestinal, such as vomiting and diarrhea, which typically occur at the beginning of treatment and often resolve on their own. The most exciting developments in pruritus management have come from our improved understanding of the specific molecular pathways of itch. This has led to the creation of highly targeted therapies. First among these is oclacitinib, sold under the brand name Apoquel. Oclacitinib is a Janus kinase, or JK, inhibitor. It works by directly blocking the JK-STAT signaling pathway inside cells, the very pathway that interleukin 31 and other key itch cytokines use to transmit their signal. Because it hits this central hub of itch signaling, it is incredibly effective and, most importantly, incredibly fast-acting. Oclacitinib can significantly reduce pruritus within a matter of hours. This makes it an outstanding choice for both stopping acute flares in their tracks and for safe, effective long-term management. It's an oral tablet, typically given twice daily for the first 2 weeks, and then reduced to once daily for maintenance. It is generally very well tolerated, with uncommon and mild side effects. The second revolutionary targeted therapy is locavetmab, which you may know as Cytopoint. This is a completely different type of treatment called a biologic therapy. Locavetmab is a caninized monoclonal antibody, a highly specific, engineered antibody that is designed to do one job and one job only, to find and neutralize circulating interleukin 31, the master itch cytokine we've been discussing. By taking interleukin 31 out of circulation, it can't bind to its receptors on nerves, and the itch signal is effectively cut off at the source. Like oclacitinib, locavetmab works very rapidly, often providing relief within 24 hours. It is administered as a simple subcutaneous injection by a veterinarian, and its effects last for 4 to 8 weeks, making it a wonderful option for long-term management, especially for owners who might struggle with giving daily pills. Because of its incredible specificity, locavetmab has a remarkable safety profile. It is a protein that is simply metabolized and broken down by the body like any other antibody, with minimal impact on the liver or kidneys, making it an excellent choice for dogs of all ages, including those with other concurrent health issues. We've journeyed from the simple definition of an itch to the complex immunology and neurobiology that drives it. We've seen how elegant scientific models and powerful new technologies are giving us an unprecedented view into the inner workings of this common affliction. It is clear that pruritus is not just an itch. It is a complex medical condition that can cause significant physical and emotional distress for our pets and disrupt the human-animal bond. The good news is that we have made enormous progress. The development of therapies that precisely target key molecules like interleukin 31 has transformed our ability to manage allergic itch safely and effectively, dramatically improving the quality of life for countless dogs and the families who love them. The future of veterinary dermatology is bright, and it's a future with a lot less scratching. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging,